Thanks a lot for spending your evening here with me today. I wanted to tell you about some of the work we've been doing in our lab, which is pretty diverse actually, but today I wanted to focus specifically on what we can do with bacteria. And so these are all pictures of different pathogenic bacteria, and we're going to talk about how some of the new tools of synthetic biology may allow us to use these bacteria for good purposes, and also to think about developing new ways of detecting and destroying pathogens that cause human disease. That being said, I think the bigger picture, and the thing that really excites me personally as somebody working in this field, is just this general technological trend that we're experiencing in our ability to engineer biological systems. I think at a very high level, what we're seeing in this area is that we're moving from kind of old school techniques that were very hard and very bespoke to be able to modify and understand biological systems. And with the advent of a lot of new technologies, we're moving to a place where we can really read and write genomes at much higher precision, fidelity, and speed than we could before. And that's really been a very exciting part of this field, in that every year, our ability to modify and understand biological systems improves. And this is really a technologically driven shift that gives us the ability to really dream about what we could do with the power of biology. So at a very basic level, as a kind of naive engineer coming into the field of biology, the way I viewed the world was like this. We have hardware, which is like the cells that we encounter, bacteria, human cells inside of our body. And what powers what these cells do is the internal software, right? So just like you can write code for a Windows or a Mac computer, we can think about programming the DNA that goes into any one of these cells. And by doing so, we can confer new functions on those cells. And in a larger sense, this is really a technological shift in our ability to engineer biological systems. So I'm sure a lot of you have heard the term genetic engineering, which 30 or 40 years ago was really transformative in our ability to get biology to do stuff for us. And what genetic engineering really introduced was this concept of being able to cut and paste DNA from one place to another. So I could take the gene that codes for insulin, and I can cut it out from its native context, and I can plug it into a bacteria and have that bacteria produce insulin for me. That's really been transformative and has launched a whole slew of new life-saving therapeutics and whole industries around this concept. What's really changed over the last 10, 15 years is that we can not only cut and paste DNA, but we can now literally go into our computers and write DNA. And that is getting cheaper and faster every year. So here's just an example. So shown here in the blue trend is basically Moore's law. So Moore was one of the founders of Intel, and basically said that there's this law in the semiconductor industry that every year or two, you can fit twice as many transistors on a given chip. And as a result, we have things like cell phones these days. We have really powerful computers, because every couple of years, we can double the computing power that we have. And so this is the trend of Moore's law. This is an exponential graph, which means that every year, we're kind of exponentially increasing our ability to program electronic systems. Now, if you look at DNA sequencing or synthesis technologies, in many cases, we're actually matching that exponential speed or even exceeding it. And that suggests that our raw ability to program biological systems is really increasing every year. What this is leading to is really democratization of biological engineering. So this is obviously an extreme example, but you can go onto your computer today. You have some idea for a piece of DNA that you want to order, some software that you want to program into your cell. I want to make a green bacteria. You can just load up a web browser, go to any one of your favorite DNA synthesis sites, type in that sequence that you want. That company will use a variety of different methods to make DNA. One of the methods that people actually use is very similar to inkjet printing, where you can basically print DNA on a chip. Then they package this information to you, put it in a FedEx envelope, ship it back to your house or to your lab, and you can plug that into a bacteria and basically test for function. Right? And so we're no longer constrained by our ability to manipulate DNA, and it's becoming easier and easier to do this. And as a result, there's been a whole slew of engineers that have gotten really interested in how we can program biological systems from the ground up, where the ultimate fantasy for some of the engineers is to be able to go in, design some genetic information, plug that into a cell so you can make devices or circuits that, based on the DNA, perform some useful function. And then eventually, can we scale up the complexity of the systems we build to something like a really complex system, a multicellular system like a tree or some complex organism that performs a defined function. Now, why might we want to do this? On one hand, it's just intellectual curiosity. We want to understand how is it that biological systems are put together. I think fundamentally, that's a very interesting question to ask. But there are a whole range of applications that people are trying to pursue with this type of technology. I'm going to focus today primarily on microbial applications, just because of time. But people are very interested in using the ability to program biological systems to treat things like cancer. Can we understand things like neurological diseases better? Can we correct genetic diseases? Can we go into a cell, modify the genetic basis of that disease, and cure it? And on the industrial front, there's been a lot of effort in thinking about, can you use this technology to make new materials? You know, biology makes a ton of wonderful and amazing materials. Can we actually reprogram cells to make new materials for us or make more of it? And in the industrial side, there's been a flurry of activity over the last 10, 15 years in this area of biofuels. Can you actually engineer cells to make interesting molecules, you know, diesel or gas or other, you know, higher value commodity chemicals? All of these are enabled by our ability to program biological systems. Now, as I mentioned, I'm going to focus on several focus topics, but we, in my lab, are very interested in a lot of applications of this technology that span what I talked about before. Today, I'm going to really focus on this concept of engineering cells to compute information. So can we turn a living bacteria into like a mini calculator? And what can we do with that power? Secondly, I'm going to talk about how we can develop new therapeutics, new infectious disease treatments for antibiotic-resistant bacteria, and how we can also use a very similar aspect of that technology for diagnostics. Can we sense the presence of pathogens more rapidly, and can we use that to proactively control how infections affect human health? But on the other front, we're really interested in understanding human diseases, building things like living materials, cells that actually replicate and make new materials for us, and engineering things like the human microbiome. So I'll start off and tell you a little bit about the work we've been doing on engineering cells to compute. The idea here is to take a cell and to stop thinking about it solely as kind of this free living thing that's trying to replicate, but trying to insert information, new software into that cell, so that cell can compute some information for you. So think about the cell as a computer. What would you want it to do? Just like our computer we have in front of us, we want it to be able to take in information. You know, we want it to be able to sense the environment. You want to be able to process that information, so compute some information, maybe store it in memory so you can remember it or report on that information at a later time. And then perhaps take that information and then respond, either by reporting to the outside world that you found the presence of some disease, or trying to produce some molecule to treat some type of disease. And so there's been a lot of interest in designing cells that can compute information. And in our lab as engineers, we like to think about this as going from concept to design and then implementing this in a cell, really thinking about forward engineering of living systems. So why might this be useful? Here's like a hypothetical example. You might think about engineering a cell that is a probiotic, similar to the bacteria that you eat in your yogurt. And that bacteria might be able to go into your gut and sense the presence of some disease that's buried deep inside of your gut that normally you have no idea is present. And so can we design a bacteria that you can eat? It goes into somewhere in your colon and senses presence of colon cancer, inflammation, or other pathogens. And then can somehow report on that information. It can maybe send a signal to the outside world saying, hey, look, I found this inside of the body. You should do something about it. Or perhaps even make a therapeutic molecule to try to treat that disease at the point of where it detects. And so this is just one idea of how you might be able to engineer cells with sense and respond capabilities for human therapeutic applications. So what do we need to do to actually make cells to do this? Well, we need to make cells capable of sensing information and understanding what that information means. So for example, we need a cell to be able to go into the gut and be able to say, if I detect the presence of this molecule and this molecule, I know that indicates it's a biomarker for colon cancer or inflammatory bowel disease. So that cell needs to be smart enough that it can take that information in, process that information, and understand it in a basic way. So we've been developing a variety of ways of making cells to be able to compute information. I'll give you one brief example here of how this could work and how this does work in certain types of engineered bacteria. So here's one type of computing circuit, okay? So this is what's known as an AND gate. It basically performs a very simple function. It has one output and two inputs, and these inputs can be arbitrary. It could be temperature and pH, or some biomarker that's related to colon cancer. And what the result of this circuit is that it only has an output that's true if both inputs are true, right? So an AND function is basically only true if all the inputs are true. And this is represented here as a truth table. So 1 represents true, 0 represents false, and the output is only true when both inputs are true. So how do we start thinking about building circuits like this inside of a cell? What we can do is we can design a bunch of very simple parts. You can think of these somewhat akin to like little Lego bricks of DNA that you can put together inside of a cell. And that gives that cell the ability to compute information. So here are some of the parts that we use. This is known as a promoter. It's basically a piece of DNA that turns on the expression of another piece of DNA. Okay, so this is known as a promoter because it promotes the production of that piece of DNA. There's another type of DNA called a terminator. A terminator basically acts as a stop sign. It basically stops the activity of a promoter, right? So these are basically you have the ability to turn something on with a promoter and stop it with a terminator. And using these simple parts, we can basically build all sorts of possible combinations of these parts together, and we can sandwich them between these symbols of DNA here. I'll show you what these do in the next slide. But basically, these are known as recombinase sites. They basically allow these parts to be flipped upside down or upright, okay? And that will allow us to compute information inside of a cell. And then what you can do is you can take this limited bag of parts, and then you can basically assemble it into a piece of DNA. Again, we can order this DNA online, or we can build it in the lab very easily. And we can then put this onto a plasmid and plug it into a bacterium so that that bacteria now has the ability to perform this basic AND logic function, where the output is only true if both of the inputs are true. And I'll show you how this works in the next slide. Here, the output is represented as this gene GFP, which basically turns the bacteria green. But this doesn't have to be GFP. It could be some therapeutic protein or some other reporter that can report this information to the outside world. All right, let's see what happens. So this is basically the circuit that we showed you before. I have a promoter that will turn on GFP expression, right? So this promoter will basically turn on the expression of an output. But the problem is, in between the promoter and the GFP, there are two stop signs, two terminators. So in the absence of any input, these stop signs basically block the activity of the promoter, and you basically get no GFP produced. The cells are basically off. And so the way we quantify this is we basically count the number of cells that express GFP or not. And by doing so, we can know whether the population is on or off. So in the presence of no inputs, both these stop signs are active. All right, so now what do? We basically use a class of proteins. They're known as recombinases. And what they do is something very cool. These proteins can recognize a specific sequence of DNA, and they'll basically flip that DNA upside down, right? So if you think about DNA as like a long string, what these recombinases do is they go in, they cut a piece of DNA out, and they flip it so it's backwards, okay? So it's a very simple operation. And what we can do is we can express these recombinases in the bacteria only in the presence of a specific input. So again, this input is arbitrary. It could be some molecule that represents some condition that you care about, like a disease. So for example, in the presence of input A, we express the red recombinase. And what that red recombinase does is it recognizes these red triangles, and it will basically flip that terminator upside down. So the resulting piece of circuit looks like this. You have one promoter, you have an upside down terminator, and an upright terminator. When the terminator is upside down, it basically is inactive. It doesn't work as a stop sign anymore. So in this case, we have one promoter that's working, we have a GFP as an output, and we have one terminator that's working and one terminator that doesn't. And so this remaining terminator is sufficient to block the activity of the GFP, okay? So in the presence of one input, there's no output. The GFP does not turn on. Now we can build another circuit that responds to a second input and expresses a blue recombinase, which flips the second terminator, right? So this is the same effect as before, where we still have one stop sign that's working, one terminator, but the other terminator is now upside down, and so GFP is no longer expressed. Now what happens if we have both inputs? Let's say this cell detects something inside my gut, and I want it to now respond. Well, in the presence of both inputs, both of these recombinases are expressed, both these terminators become upside down, they no longer work as stop signs, and as a result, GFP can be expressed by the promoter. So this promoter reads the GFP, and that's the output, and if we look at the number of cells that now express GFP, it suddenly becomes high. So with a very simple genetic circuit that looks like this, it takes you just a few days to build this. You can literally go online for a few $100. You can order this sequence and plug it into your bacteria of interest. You can perform a very similar function, a simple function, which basically is an AND gate. It takes two inputs and performs a logical function. Now, for the people who are interested in computation, there's basically 16 variations on logic that you can do on two inputs. And so, for example, we've showed that we can build all possible 16 two-input logic gates using this approach. You can build things like AND gates, OR gates, which basically do OR instead of AND. You can build a bunch of other gates that basically enable you to perform arbitrary computation inside of a cell. And so what this is really doing is imparting some very simple but useful intelligence into the cell and getting the cell to basically respond to an environment and basically produce a molecule that we care about. In the bigger picture, I think this is just one example of where synthetic biologists are very interested in building new types of therapies using this type of approach. where you can imagine building circuits. So this is an example of a genetic circuit. These are genes like I showed you before, and they interact with each other in some complex way. You can plug these circuits into a cell. So this cell could be a bacteria that you eat as a probiotic. It could be a cell inside of your body that you can deliver this genetic circuit into that cell. And when that cell can then go into the patient and recognize malfunctions in the native wiring of some of our native functions that causes disease. And the idea is if you can interface these synthetic circuits and somehow basically correct this dysfunction or sense a response to dysfunction inside the cell, we may have a new way of building new therapeutics that are more intelligent than the ones that we've done before. So that's one example where I think bacterial and synthetic biology can be quite useful for building new therapeutic agents. And I'll give you another example where the tools of synthetic biology really allow us to target another class of infections, of diseases, antibiotic-resistant bacteria. So I'm going to talk about two brief stories. One where we've used very similar technologies, the ability to program DNA. And by doing so, we can make very targeted therapies for antibiotic-resistant bacteria. And I'll tell you why we might care about that. And secondly, how we can use the same technology, but just modify it, to build systems that actually act as very rapid microbial diagnostics. So being able to detect the presence of a pathogen is sometimes just as important as being able to treat it. And so we can do both of these things quite readily now with some of the tools. All right, so if you pay attention to the news, actually this is very timely because last week President Obama actually announced new initiatives to try to combat antibiotic resistance. And the UK has also put this really high in their priority list of really emerging human health applications that we need to take care of. But so here's the basic fundamental problem. There's more and more resistant organisms, bacteria, in the environment and in our hospitals. So this is MRSA, methicillin-resistant Staph aureus. It's one type of antibiotic-resistant bacteria. And you look at the trends over time, we've gotten kind of increasing numbers or increasing incidence of antibiotic-resistant infections. And at the same time, the number of new antibiotics that are being developed by the pharmaceutical industry and being introduced into the market is decreasing. And these two trends are obviously not good with respect to each other. So some of the organisms that we quite care about are highlighted here. The CDC actually released a threat report last year focusing on specific organisms that they think are very urgent or serious threat levels. So these are two types of bacteria. These are all related to E. coli. And they basically have very different resistance mechanisms. One basically is very resistant to a class of drugs known as carbapenems. And this bacterial type has been classified as urgent because it's almost resistant to almost every antibiotic that you might want to use to treat this type of infection. And these are some of the statistics that the CDC has reported on these types of bacteria every year. There's another type of bacteria, these extended spectrum beta-lactamase producing bacteria that are also quite widespread and cause a very significant burden on our health system. Now, why do we get antibiotic resistance? Well, we are treating bacteria with antibiotics all the time, and naturally the bacteria want to find a way to survive. The way I like to think about this is the antibiotics we have are very broad spectrum. It's like trying to use napalm against a huge community of different bacteria. When we take antibiotics, that single antibiotic doesn't just kill the pathogen, but it kills and affects even the good bacteria that's living inside of us, right? There are trillions of bacteria that live in our gut. Most of them are doing good things, not bad things. But when you take an antibiotic, you're basically blasting the entire population with this threat. And as a result, these bacteria have the pressure to basically evolve resistance. They can do this in a variety of different ways. They can basically find mutations inside of their genes that leads them to become resistant. Once they've done that, they can also transfer genes, so bacteria actually share information with each other. So bacteria can pass DNA from themselves to their friends. And for example, they can pass resistance genes to each other to increase the resistance of other bacteria. The other problem this can cause is it causes overgrowth of bacteria that cause pathogens, that cause disease. So for example, Clostridium difficile is a type of bacteria that causes very serious diarrheal infections when you take antibiotics and you kill off all the good bacteria in your gut. And so the question we have is, can we actually go for a very different way of treating infections? Instead of trying to blast the population and kill off the good bacteria, can we make highly targeted antimicrobials that specifically recognize bacteria that carry a certain signature and only kill those bacteria and don't affect everyone else? And so we've been very fortunate that there have been recent tools that have been developed that allow us to go into bacteria and go into a wide variety of organisms and actually specifically target DNA that we don't like. And so here's one example of these systems. This is the CRISPR system. It's basically, you can conceptualize the system as like a scissor that goes in and cuts DNA. So this CRISPR system produces a protein known as Cas9, and this Cas9 protein will go in, find a piece of DNA that we don't like, and cut it. And by doing so, it can disrupt that gene. Now, you can tell this scissor to go anywhere you want in a bacterial genome by basically expressing what's known as a guide RNA, shown here. So these RNA sequences that you produce inside of a cell basically act as like a little homing address that tells that scissor where to cut. And so this is a very efficient way of trying to discriminate bacteria between each other, because it allows you to say, only cut bacteria that have a specific resistance gene versus bacteria that do not carry that resistance gene. So how can we use this type of tool, right? So the idea is we want to take these scissors, we want to deliver them into bacteria and only cut DNA in those bacteria that carry bad genes, genes that cause resistance or genes that are virulence causing, cause disease in a human. And so one way of doing this is to basically load this CRISPR scissor system into bacteriophages. So here's an example where we've taken a cell, we've encoded the CRISPR system, so this is again a short piece of DNA that we can design in the lab, and we can package these into bacteriophages. Now bacteriophages are basically bacterial viruses. They're basically these small nanoparticles that are produced by bacteria, and they basically target bacteria, so they can bind to bacteria and they inject this information, this DNA into bacteria. So we like to think about them as kind of like bacterial mailmen, right? They go around, they find bacteria, and they deliver this genetic information into the bacteria. And we can modify these bacteriophages arbitrarily to have them deliver any piece of DNA that we want. And in this case, we're doing that with these little scissor proteins. And so the idea is when we do this, the bacteria inject the scissors, the CRISPR system, that forms this complex that now can go search for genes that we might want to target, right? So I'll give you one example of where we've used this to get very specific antimicrobial targeting inside of bacteria. So here is an example where we want to take bacteria and we want to target bacteria that only carry a specific resistance gene. Okay, so we compare two different bacterial types. One bacterial type is known as wild type. These are good bacteria. They don't have any resistance gene. So that's shown here. WT means wild type. And then we take a very closely related version of this bacteria, but we introduce a very small mutation in one of their genes that is sufficient to confer resistance to antibiotics to this bacteria. So this very tiny, there's essentially just a few base pairs of DNA difference between these two different types of bacteria. But the bacteria on the left is sensitive to antibiotics. The bacteria on the right is resistant to antibiotics. So now we want to develop a system that will only kill the bacteria on the right, but not kill the bacteria on the left. And so the way this works is we design these CRISPR systems to be packaged into these bacteriophages. These phages will inject the scissor system, and the scissor will only detect the presence of this very specific mutation that confers antibiotic resistance. When it does that, it cuts the DNA, and bacteria clearly don't like their DNA being cut. It disrupts their ability to use that DNA as information, and that leads to cell death, all right? And so here's just some data showing that this works. So we can take, for example, these wild-type bacteria. We can apply these bacteriophages, and when we target just the wild type, these are the good innocuous bacteria, we basically get no killing. So you compare, for example, black versus gray. Gray is where we add the specific bacteriophage here. We basically don't see any change in the number of viable cells, right? So this is, on the y-axis, the number of cells that we have, right? So we have about almost 10 to the 8th bacteria, about 100 million bacteria living in this population. Now, in the presence of the bacteriophage, you get a significant drop in the survival of the pathogenic bacteria. So using the same exact bacteriophage, shown here in gray, we don't kill the wild type, but we can kill the antibiotic-resistant version of that bacteria very effectively. In a sense, we get over 10,000-fold killing of the drug-resistant bacteria. So if you compare this data point to this data point, we've dropped from about 100, almost 100 million bacteria down to somewhere around 1,000 or so bacteria. So it's a very significant fold killing that you get. And the reason why this works is because you have this very specific scissor element that allows you to recognize and distinguish bacteria based on the genes they carry. So now we can design basically antimicrobial agents that are not targeting everybody, but only targeting bacteria that carry those bad genes that we want to get rid of. So here's one example where the tools of synthetic biology allow us to very easily reprogram these scissors to target any target of interest. I'll give you another example where we can also use bacteriophages as a way of breaking up bacteria that live in what's known as biofilms. So here's another case where we can use the same technology, the ability to reprogram these bacteriophages to disrupt basically this bacterial city that they live in. So, classically, if you've taken high school biology, you basically remember bacteria are living like liquid, right? We think about bacteria as free floating around. That's actually not the dominant way that bacteria live. Most bacteria are like humans. They like to have a home. They will stick to a surface and they'll build a community for themselves. So here's an example where bacteria can float around. This might be in the environment. This might be in a person. They'll stick to a surface and they start building these three-dimensional communities of bacteria. These are known as biofilms. And these bacteria live in these communities and they make kind of a house for themselves. They'll make all sorts of extracellular materials that protect themselves from the outside world. They protect themselves from antibiotics. They protect themselves from the immune system, et cetera. In many sense, you can think about this kind of like fruit jello, where the bacteria are like the fruit inside that fruit jello, and they're making all sorts of like gelatinous stuff on their outside to stabilize the community in which they live. Now, this is a problem for all sorts of different infections. So if you don't brush your teeth for a few days, you'll know this quite readily. You can scrape plaque off your teeth, and that's basically a form of bacterial biofilms. They stick to that surface, and they form that plaque. So dental infections significantly caused by biofilms. Cystic fibrosis is another example where bacteria will live in the lung of patients that can't clear mucus very efficiently. And they'll make these very intractable houses for themselves that are difficult to treat. With things like medical devices, bacteria love to stick to these surfaces. They're kind of artificial surfaces. They can grow really efficiently on them. And then in the industrial setting, this is actually a problem too. So you may not have thought about this, but like the HVAC systems that are powering the air conditioning in this room has two loops. They have a hot side and a cold side, and they transfer heat from one place to another. That's the way the air conditioning system works. And biofilms love to live at that interface. And what they do is they form these layers that basically insulate that heat transfer. And as a result, the efficiency of those systems drops significantly. So people currently dump all sorts of like toxic chemicals into the HVAC system to try to treat these biofilms. In food, and I'll talk about food a little bit later in this talk, biofilms like to form on all sorts of surfaces. So you probably remember the E. coli spinach outbreak, maybe 9 years ago, where biofilms are stuck to surfaces and very difficult to clean off. And so one of the ways we can actually design bacteriophages to treat this problem is to turn, again, using the bacteriophage as a mailman for delivering genetic information to a biofilm, we can program these bacteriophages to basically trigger biofilms to destroy themselves. So here's the idea. Here we have biofilms sticking on a surface. These bacteria build communities. And again, if you think about this, the bacteria that are living on the bottom of these communities are actually pretty happy. They're kind of shielded from antibiotics or immune cells that might be trying to invade them from the outside world. And similarly, if we just put bacteriophages on their own onto these biofilms, the bacteriophages find it very hard to penetrate very deeply into the biofilm, which means that you can kill the kind of the top layer of these things, but you can't really penetrate very deeply. So how do we get down deep and kill those biofilms that we want? One way of doing that is to actually engineer these bacteriophages to express biofilm-degrading enzymes during infection. So again, we're going back to this idea of ordering DNA. We can design DNA, we can plug it into the bacteriophage, and as a result, when the bacteriophage infects the bacteria, it gets the bacteria to produce these enzymes inside of the bacteria. So here we have enzymes shown in red and bacteriophages shown in black. They're being produced inside of these bacteria. And when the bacteria explode, what you get is you get release of these bacteriophages, so it allows these bacteriophages to go, they can make more of themselves and penetrate deeper and deeper into the biofilm. At the same time, you produce these enzymes shown here in red. And what these enzymes do is they chew up that jello matrix, right? So these bacteria are producing things to protect themselves. These enzymes will chew up that matrix and basically allows you to disrupt the biofilm from within itself. We like to think about this as a Trojan horse. You're kind of using the bacteriophage to sneak this Trojan horse into the biofilm, and then all sorts of like little guys pop out, make more of themselves, and destroy the city from within, right? So this is a very similar concept, and we've shown now that you can use this technique to get many orders of magnitude, several thousand or 10,000-fold destruction of biofilms using this approach. So I wanted to introduce this because it's an example of where some of the technologies that we've been developing in the lab are starting to make it into real-world applications. And so this previous work that I showed you here was actually done during my PhD. I did this with Jim Collins over at Boston University at the time. And we were quite interested, now that we had this new technology, on trying to develop it for the outside world and trying to use it for actual human applications. And so we've published a couple papers on this, and as academics, we were actually quite excited. It's always great when you have a publication. We talked about using enzymatic bacteriophages, as I showed you in the last slide, to break up biofilms. We also developed other bacteriophages that I don't have time to talk about today, but basically these bacteriophages will go into bacteria and knock down specific resistance genes and allow you to synergistically work with antibiotics. And so we published these 2 papers, and we were quite excited. We wanted to try to translate this into the outside world. So this next part, the last kind of part of my talk, is really about the trials and tribulations of trying to take some of these technologies into the real world. Fortunately, at the time, I was in the lab of Jim with another colleague of mine, Michael Karras, who was really excited about trying to bring some of these lab technologies into the real world. And so after a lot of hard work, we ended up spinning this technology out into a company now known as Sample 6. And what we've done is to actually, so what we did initially as very naive grad students was to basically try to find ways of funding this research, right? We wanted to take the next step, and our idea was really set on therapeutics, right? I was trained as a medical doctor, although fortunately for patients, I don't actually see patients because I'm a very bad doctor. But we wanted to have this impact patients. And so we wanted to develop therapies. And so the way we thought about doing this is instead of trying to raise VC money, at the time, this is like 2008, 2009. This is actually like the worst time in history, one of the worst times in history to raise VC money. So we bootstrapped our way up there. We got pretty creative. We actually realized that there are tons of business plan competitions that are open at a lot of schools. And kind of the loophole that we found is that you don't actually have to be a student at any one of those schools to win the money. So as you can see here, we went to Purdue, UT Austin, all these different places. We did pretty well. We actually raised about $100,000 through that approach and managed to score ourselves with very tiny lab space. Actually, this is a lab at UMass Boston, just down near JFK Library. We actually only got half of this space because we didn't have enough money. So this is a little bit overstating how the company started. We got the left side of this picture. And we managed to actually start generating some interesting data for our own, and we were really excited about the application of this for real-world therapeutics, right? And so we eventually managed to also get some support from Boston University and SBIRs, and then we ran into a big hurdle. This big hurdle was that the regulatory issues are very challenging for new therapeutics, and I think as an academic, this is understandable, and we're continuing to push on these types of technologies for human applications. But just to put this into context, no phage therapies currently exist in the Western world. Phages are actually very fascinating history. If you have time to go back and look at this on Wikipedia, phages were discovered like 100 years ago and actually were used as antimicrobial therapies before antibiotics came around. But after antibiotics came around, they kind of really fell out of disfavor. They were never really adopted in Western medicine over the last 50 years. Even though like in places like Republic of Georgia or Eastern Europe or Russia, they continue to actually make phages for therapies. And actually an interesting anecdote is you can go to the Republic of Georgia, go to a pharmacy and actually buy like a vial of phage over the counter and drink it if you want. So, the regulatory hurdles for this are very different depending on where you are. The challenge for us is, if as a small company, you want to try to develop, something that you think has a lot of promise and take it through the FDA. Some of the estimates are like it takes over $1 billion in 10 years to do this. And for us, it was very challenging at the time. So we basically went back to the drawing board and tried to figure out where this technology could be reapplied. And so that's what I'm going to tell you in the last few minutes of this talk. Where we've ended on this is actually using the technology for microbial diagnostics. And so at the time, we didn't actually think about this idea because we were really focused on therapeutics, but you can actually repurpose bacteriophages to light up bacteria. And so I'll tell you about how this can work. Now, the number one application that we're going after for now is this area of food safety. And so as a basic scientist, we often don't think about food safety that much. We think maybe it should be a solved problem, right? People don't get sick that often from food. But actually, if you look at the statistics, we still have about 5,000 people who die every year. A lot of people get sick, so you probably know somebody every year that gets some sort of food poisoning. Globally, obviously, this is much more of an issue. And aside from just the human health costs, there's a lot of significant economic costs to these food safety issues. You might be very surprised to find, though, that pathogen detection techniques, so the techniques that the food manufacturers use to detect whether their food is clean or not, often takes days to get results. And that means that, let's say you go out for lunch, you're going to buy a salad. The guy who's making your salad, well, that salad goes bad in a few days. They're not going to wait in their factory to find out that that salad is clean before shipping it out, right? They're losing hard dollars by doing that. And that's why oftentimes you get the situation where people are sending out food, and a few days later you're hearing about these massive recalls in the news that we find contamination, we've got to bring everything back, at which point it may be too late for certain people, and it's also very expensive. This is something that I think over the last 9 or 10 years has really risen in the public's awareness 2006 spinach recall was a big problem. It actually caused a significant drop in spinach production and spinach consumption in the United States. People died. There was significant costs for these recalls. So this spinach was contaminated by E. coli. Peanut Corporation, this is actually really timely. So the Peanut Corporation actually had a salmonella outbreak. The CEO and the food safety manager of this company actually were on trial, and they actually just got convicted this past week for not basically keeping a very clean plant. And that was they were found guilty on criminal charges, actually, so this went beyond just civil charges. This was salmonella and peanut butter. We had the cantaloupe listeria outbreak a few years ago that you may have heard of. And one of the scary things is, you sign up for the FDA food recall list, which you may or may not want to do after this talk. But if you do that, you basically find out that every major food manufacturer and every major even retail place has had a food recall in a very short period of time. So in a single quarter of last year, or 2 years ago, there were 550 food recalls. And this is an issue that happens across a wide variety of food types, yogurt, chocolate, dog food, et cetera. It's a global problem. We think that this problem is probably getting worse and not better. There's a very globalized food chain these days. Oftentimes, I didn't realize this again until we got into this industry, but the food chain is so globalized that oftentimes the manufacturer they don't actually know necessarily where a specific thing came from. It came from such a large distribution chain. People are getting lazier and basically like to eat ready-to-eat foods. We see pre-washed, triple-washed lettuce or in the supermarket, and we go home and we don't wash the lettuce. Things like social media and Twitter and things like that are making food outbreaks, information about food outbreaks becoming more widely available. So how can we use synthetic biology to solve this problem? And so the trick here is really using that. Again, we're going back to that bacteriophage, our trusty friend. We're going back to them many times in this talk today. We can use them as microbial detectors, and here's how we can do that. Bacteriophages are actually very highly discriminatory elements. A single bacteriophage will recognize a specific type of bacteria. You can't find a bacteriophage that targets all bacteria. There's no such known superphage that targets everything. And so as a result, phages can be used to distinguish between different bacterial types. So a given phage might target listeria, but not target E. coli. I can find a different phage that targets E. coli, but doesn't target salmonella. And so by assembling a cocktail of bacteriophages that has the specificity I want for that pathogen, I can get very specific detection of certain bacterial types. So that's the specificity side. That's how I distinguish different bacteria from each other. Now, how do I light up the bacteria, right? Bacteria are largely invisible to the naked eye. How do I get the bacteria to make a signal that I can detect? Well, we can take the bacteria and we can modify bacteriophage, we can modify that genetic information. Instead of delivering the bacteriophage genome, we get it to deliver a piece of DNA that expresses an enzyme. So here you see bacteriophages injecting the DNA. The DNA produces a lot of RNA. RNA produces a lot of proteins. And what we do is we make those proteins light-producing enzymes. We basically take enzymes that you find in like fireflies, and we basically put that into the bacteria, so the bacteria becomes like a little light bulb. It basically produces light when it's present and when it's been infected by the bacteriophage. And what this allows you to do, as I'll show you, is that the phages can very rapidly target bacteria and produce light in a short period of time. So you don't have to wait several days to get your answer. The reason why this works is because biology provides like multiple layers of signal amplification. So from a single bacteria, you want to produce as much light as you can, because that allows you to get very sensitive detection. And the reason why this can happen is because from a single cell, we can get a bacteriophage to infect, we can produce lots of RNA. Each one of those RNAs produce lots of protein as enzymes, and each one of those enzymes basically makes tons of photons. And this gives you a significant amount of signal amplification that allows you to detect the bacteria that are present. Now, so here's an example. This is obviously a very extreme example. In most cases of food are not this extreme. But here's basically on the left, bacteria present, versus here on the right, no bacteria that are present. And basically by looking at the production of light, we can tell whether bacteria are there or not. So it's a very simple assay. And so here's what this enables you to do. There are three kind of existing classes of therapy, diagnostics that are on the market currently. The classic is culture. So this is going back to classic microbiology. You just basically see if the bacteria grows. For something like listeria, that takes many, many days because listeria grows very slowly. So basically a lot of these food companies will take the bacteria, they'll grow it up, and then they'll wait to see if bacteria shows up. That takes a long time. More advanced assays, so for example, immunoassays, that's basically like the pregnancy test. It's an antibody that binds to a specific target. those antibodies are very, very specific. So they're great at discriminating between different bacteria, just like the phages. The challenge, though, is that those antibodies don't have the same level of signal amplification that we can get with a bacteriophage. And as a result, they're not very sensitive. So what happens is if you take a sample, you need to grow up more of that bacteria to get enough bacteria that you can detect it. And so again, the reason why this takes a while is you need to do that what's known as that primary enrichment step, where you're growing the bacteria. A lot of the food plants don't like to grow the bacteria in their plant for obvious reasons. And so they ship off these assays to an off-site laboratory, and so just FedEx time takes an additional amount of time. More recent technologies like PCR. So PCR is a molecular technique that allows you to amplify the DNA signature of these bacteria. That's very sensitive. You can get lots and lots of amplification of the DNA signature, but it's very challenging to make PCR work in very complex environments and also make it very specific. So again, PCR is very sensitive but not very specific. You need to grow up a lot of the bacteria to be able to do the detection. And so the difference with the bacteriophages is that we've kind of forced a lot of the intelligence of the assay onto the phage. It basically allows you to do very specific detection and a lot of amplification, and to get to the point where you can actually do this assay on site. We think this is a broadly applicable platform where we're starting off with food safety, which is a very large and important area, but you can imagine applying this for other settings. In clinical settings, it might be useful to very rapidly know what bacteria that you're coming in with. Industrial settings, again, you might want to know the bacteria that are there. So here's different food types. So the three major pathogens of food in the United States are Salmonella and E. coli and Listeria. So these are the three major ones that we want to go after. And what we want to do is to make a test easy enough that anyone can use it. So the joke around the company is that we want to make a test easy enough that the CEO can run it. And so the idea is we want a very simple test. Here's a little sponge. So basically the way this test works is you take the sponge out, you wipe a surface, right? And you test whether that surface has bacteria or not. You put it back into this little bag, and then you pour the little phages in. You let this sit there for a few hours. You take the liquid in that phage, and then you basically look at it in this little machine. It's a luminometer. Basically detects the presence of light or not. It's a very sensitive way of detecting that. And so this assay is very simple. It's like you can sponge it. You don't need any training. There's very little processing steps. You just kind of sponge, pop it in a bag, and read the light that produces at the end of the day. What this allows you to do is to basically sample very proactively, right? So we can get to the details of this later, but there's two types of kind of food safety testing. One is basically environmental testing, where you want to make sure the factory itself is clean. And by doing so, you can have confidence that the food that you're making in that factory is relatively clean. And so you can sample a lot of different locations in that factory, and basically this allows you to sample a lot quickly. And also we've tied in the assay to the cloud so that you can upload this data very quickly and see what's going on inside of your plant. All right, so here's some data. So this on the left is Listeria. So Listeria is actually one of the very difficult bugs to detect because it grows very slowly. Listeria is especially problematic for immunocompromised people or pregnant people. So, for example, you're often warned if you're pregnant not to eat lunch meats or things like that because it can have Listeria on it. It likes to grow in cold scenarios, or just keeping something in the refrigerator doesn't necessarily inhibit Listeria from growing. Here's how the assay works. So here on the y-axis, RLU means relative light units, so the amount of light that's produced. Basically, it's a measure of how many photons that come out of this assay. And on the x-axis, we dilute down the number of cells in the given assay from 1 million down to about 1. You can't see here just because of magnification. If we zoomed up, you would actually be able to tell that the amount of signal that's produced by one cell is very close to the negative control, but it's actually statistically significant. And clearly, just above 1 cell, 2 cells, and 3, you can actually get pretty good detection of Listeria. You can do this quite quickly. So in the lab, within about 60 minutes, you can get a statistically significant detection of light production from these bacteria. E. coli, we do even better just because E. coli is much faster and easier organism to work with. So this is 600 cells here, 60 cells here in the middle, and 6 cells here. And so you can see within like about 30 minutes, you get a bump in the amount of light that's produced. That's higher than the dotted line here, which is the lower limit of detection. So that's where we can't detect anything. And here, with a lot of bacteria, you can actually even get even quicker detection, even within like 10 minutes. You can get a lot of light being produced by a single bacteria. And so the idea here is we want to be proactive about food testing, where we can provide an assay that's very sensitive and specific, easy to use, tie that into all sorts of analytics and big data solutions for analyzing like where contaminations are happening. It's been very satisfying for me because this work grew out of the PhD project, eventually made it into the real world. This is our actually certification. We just got this just a few months ago. And so this is a product that's actually being used right now in a variety of different settings to be proactive about food testing. So with that, I'd like to finish up. I want to tell you about a couple of different concepts that we've talked about today. one idea that we talked about today is this idea of really getting much more specific about how we treat infections. So instead of the current model where you go to a doctor, you're kind of sick, you don't really know what's causing it. Oftentimes you go in, you say, I have a pneumonia. They have no idea what bacteria is causing that pneumonia. And as a result, they give you a broad spectrum antibiotic. The idea is let's give you something that's broad enough that'll kind of blast everything and hopefully you'll get better. We've talked about what that can lead to. Now, our vision for the future is to change this. Imagine being able to go to that doctor, have a very rapid diagnostic test where they can take a sample and in just a few hours they can know what bacteria is the pathogenic cause. And then we can not only take that diagnostic, but we can develop and provide a very customized therapeutic that will go and treat that infection specifically. And this idea of doing very targeted narrow spectrum therapies that we can actually iterate on is one of the hopes that we have for this field. So in summary, I've told you today about how synthetic biology is an emerging discipline for engineering biological systems. It's really being driven by technology. Cells can be engineered to sense and respond. So going back to that idea of that probiotic yogurt that goes into your gut and senses information, we can load these cells with sensors to be able to do that. We talk about how broad spectrum antibiotics may not be ideal and how we can design very targeted antimicrobials with the CRISPR-Cas system to treat those antibiotic-resistant bacteria. We've talked about how microbial detection is really an integral part of not only food safety, but potentially for personalized therapies. And we talked about how engineered bacteriophages can give you sensitive, specific, and rapid detection. I think the last message I'd like to leave you with is kind of at a higher level, thinking about what we can do with this technology, right? The technology is really improving at an exponential rate, at least our raw ability to program biological systems, and really kind of opens up the imagination for what can you do, what should you do, what shouldn't you do with this technology. It's also been very interesting to be in this field because it's a very interdisciplinary set of people. So in my lab, we have biologists, we have people who come from engineering, people who come from more of the medical side of things. And thinking about how to integrate all these very different groups of people to use this technology for the future has been a very exciting path for us. Lastly, I'd like to thank the people in the lab who did the really hard work. This is the group. This is our retreat that we took to Cape Cod last month. The work that was done on the engineered logic systems was done by Piro Suti, who's now at Novartis. He was a postdoc in the group. The work that was done on the CRISPR-Cas system was done by Rob Sidorick and Mark Mimi. I think some of them are actually here in the audience and somewhere in this picture as well. And the Sample 6 team, I've been very fortunate to work with a group of 16 scientists now located in Cambridge to try to develop some of the technologies forward. With that, I'm happy to take any questions. Feel free to email me or check out some of the stuff we've been doing in our lab. Thank you a lot for your time.
Thank you for a great talk, Tim. That was really interesting. We have a few ways in which you can ask questions, and we have about 10 or 15 minutes to do that. I will put this microphone up here, and there's another one on the other side, and you can come down and ask questions, although I might ask you to move your baby a little to make that possible. If you can't get down, you can raise your hand and we'll come to you. And last, you can tweet the question at #BroadTalks, and I'll be scanning that Twitter feed. As people line up, I wanted to ask the first question, actually, which has to do with cost.
Sure.
Both broad-spectrum antibiotics are really cheap. Kind of what would it be to actually put in an actually specific one? And what is this going to do to the cost of food safety monitoring? Just some ballpark
Oh yeah,
figure.
so for the food test, actually, the raw cost for us to actually make these assays, and actually the raw cost for making a lot of drugs is actually relatively low. So for example, in the food assay, we can make each assay cheaper or about the same as kind of existing methods, PCR, immunoassay. So it's not more expensive. I think where you really get into cost issues is if you layer on all the regulatory things you need to do for therapeutics, that's really where the cost, you're really subsidizing the cost of the R&D and the regulatory path, and that's really where the cost comes from. Ideally for us, we would love to be in a situation very similar to like the flu vaccine, where the flu vaccine is updated every year to include new components. And if you could think about engineering therapies that could be replaced like that, using the same framework, that could really go a long ways in reducing some of that. But that's a very radical way of thinking about treating infections that it will take, I think, a while for us to get there.
So just kind of number, 10, 100?
So the cost for this assay is in the ballpark of like $10 or $20 per test. And that's how much it costs for other things. Yeah.
Hi, great presentation. Thanks so much. A lot of interesting applications. Two questions. One is, what was the most significant hurdle in that regulatory path? I'm sure there were many complexities, but often when you're going through the assessment, one kind of stands out as being the one that really makes you step back and say, maybe we need to find an alternative. I'd be curious as to whether there was one in particular. And then another question about the system itself. If you were to go into humans with it, is there any risk of a mutation in the targeting system itself that could then be spread so that you're no longer targeting the intended genes with the CRISPR or with the engineering? You're now perhaps doing something off target. And is there a risk of creating adverse effects from that?
Yeah, so those are great questions. Thanks a lot. So the first question was really focused around what are the regulatory hurdles for some of these technologies. And one of the major hurdles that we face is that for most phage therapies, phage-based therapies, you need a cocktail of phages to sufficiently cover the range that you want, because these phages can be very specific. So oftentimes you would think about building a cocktail of multiple 2 or 3, 4 phages together. And the way FDA often views these is that you have to get each one of the components approved on their own, and then you have to prove that you have a kind of well-controlled way of mixing them, and that just really scales the cost very significantly. And so it's in very rare cases, like with the Ebola antibodies, that you can mix polyclonal antibodies together from a process, or you can mix vaccine components for influenza or other vaccine components, that you can actually do that at a reasonable level. But with phages, there is no precedent, first of all, and secondly, we faced that hurdle. So that was kind of one of the big daunting things for us, and it still continues to be, even though there are people working on that issue. So there are phage-based therapies in phase 2 trials now in Europe that are being done. So hopefully if they make it through, they pave a way for the rest of us. The second question was really about off-target and also about the question of resistance. And so the way we view this is that I think bacteria are always going to beat us at the end of the day. They've been around in this world a lot, and they have ways of replicating very quickly and having mutations very quickly. And so I will never, in my opinion, find a therapy that is immune to the evolution of resistance. What we want to do is get ourselves in a position where if resistance happens, we can counter that resistance. It's always going to be a matter of kind of continuous warfare with the bacteria. And so the idea with some of the synthetic strategies we've developed is, and others that people are thinking about, is that when you get resistance, can you rapidly, within a very short amount of time, like a month or so, develop something that gets around that? And so I think some of the tools in synthetic biology allow you to do that just because you can iterate through designs pretty rapidly. And that's very different than discovering new antibiotics. It's very challenging to discover new ones from large screening efforts. Thanks.
And the off-target risk?
Oh, it's off-target, sorry, yeah. So the phages themselves are quite specific, and so that gives you one level of specificity. The CRISPR system, this version of the CRISPR system is kind of the first ones that have been developed. People are trying to develop even more specific versions of it that even have less propensity. Now, in our assays, we were not able to detect significant off-target effects, so we were able to discriminate bacteria that really just differed by a few base pairs of DNA. So it seemed to be quite specific. The other way to get around this is to, instead of using phages that can actively replicate on their own, and that might lead to more mutations, is to actually use phages kind of as dead vectors. So they don't actually replicate, they're not really alive, they're just delivering stuff. And when they do that, they don't really have the ability to propagate themselves and mutate. So that's another way of getting around that issue. Yeah,
Thank you.
Thanks.
Yes, your last answer kind of touches on my question. We know when agriculture introduced gene changes to enhance a crop and so on, there's been terrible resistance. One should not tinker with nature, leave it alone. And we also know that various medicines and antibiotics and so on wind up in the source stream and out into the harbor and so on. So what is the risk with phages running everywhere or these modified pieces of genes running everywhere? Are we tampering with natural evolution that took billions of years?
That's a great question. So I think this is a question that as the community of synthetic biologists, we take very seriously, because it's obviously something that we want to be cognizant of and deal with. So I would answer this in a variety of different ways. So firstly, the phages on their own are quite abundant. They're actually the most abundant particles in the world. So we're not for example, it's thought that we have more bacteria in our gut than we have human cells, and it's thought that we have more phages than bacteria in those similar systems. So phages on their own, I think, are relatively ubiquitous in the environment. Now the question really is, what happens when we start making genetic modifications to those, right? And so I think there's a big distinction, as I mentioned earlier, about releasing phages that can actively replicate on their own, that have modified genetic information, versus something that just delivers a piece of DNA and cannot replicate. Because the latter I view more as kind of a classic drug-type therapy, where you're just delivering something that has a certain effect and it goes away, versus the first one, something that freely replicates. And obviously the first one is more concerning. I think the second one is probably more likely to be palatable for regulatory reasons in the short term. That being said, there are a lot of efforts in the field to build essentially what's known as biocontainment strategies. So ways of making sure that, or at least reducing, so we can never be 100% sure, but we can reduce the statistical chance of things escaping or mutating below a very low level. And so there's a lot of efforts now to build things like safety switches inside of engineered organisms that commit suicide if they're in the wrong conditions, to build organisms that cannot freely replicate unless you feed them something artificial, or even people have kind of radically recoded organisms to have an alternative genetic code such that the software that runs in the engineered organism can never function in a natural organism because the way they interpret that software is completely different. That being said, there's no examples of, as far as I know, of kind of engineered organisms like this, other than in the agriculture space, but of more of the recent work that hasn't actually been freely released. We are working in the lab and waiting for those kind of technologies to be developed before we actually push forward with some of these more radical applications.
Okay, thank you.
Thanks.
Hello, my name is Ben. Just have a question. Do you think eventually all broad-spectrum antibiotics will become obsolete? And if we use a more targeted approach, do you think that method will not be able to address all the challenges that are not able to be addressed by broad-spectrum antibiotics? Or will there still be some problems that will persist even with a more targeted approach?
Yeah, so I kind of envision that, at least in the near to midterm, it's likely that both antimicrobial, the targeted as well as the broad spectrum, will continue to exist, both for kind of regulatory and inertial reasons, but also for scientific reasons, in that, just by the law of mathematics, if you're developing viral therapies, you have to develop a lot more of them to cover all the infections that people encounter. And so the broad spectrum has the nice advantage that you don't necessarily have to know what's going on to be able to treat it, right? I think in the very long term, it's hopeful to us that most broad spectrum antimicrobials will be replaced, except in conditions where we either don't have a narrow spectrum available, or it's just so urgent that we need to try to blast it at first. So that's kind of where we envision it, but it's kind of hard to know exactly where those cutoffs are going to be.
And then do you think the targeting will be able to overcome the bugs that are resistant now to broad?
Yeah, so I think the targeted therapies themselves, any given target therapy, the bugs can develop resistance to. So the challenge for us is to be able to develop a pipeline where we can constantly iterate and find new ways of overcoming that. And so here's just an example. During my PhD, it took 3 months for us to make a single bacteriophage. The company now can make like 14 different new bacteriophages in a week. And so that cycle is completely different, and it really changes the way you think about how quickly can you keep up with the bacteria. And it's, I think that's the paradigm we're shooting for. Okay. Yeah, thanks.
Awesome.
What phage therapies are available in the Republic of Georgia, and do they work?
It's a great question. So you can basically go there, and we have some collaborators who actually brought us a couple vials. They're little things you just drink. So they target things like Staph aureus. So they don't target specific organisms there necessarily. They basically sell them for specific conditions. Like you have diarrhea or something, you take this. The challenge with phage therapy, it's actually really interesting history. A lot of the studies that have been done in other parts of the world or very early on in the discovery of phages were not up to the standards of what we would expect in like a double-blind, placebo-controlled, clinical trial. And a lot of the reports have been anecdotal. So it's very hard for us to know exactly, at least from our perspective, whether a lot of those therapies, how efficacious they are, especially versus other kind of gold standards. What we do know is that there have been phase 1 and phase 2 clinical trials in Europe, and they've looked at how effective these are at killing, for example, Pseudomonas in ear infections, and they seem to be pretty effective in those settings. And so we're starting to get some data that's a little bit more rigorous, but I would say that we have a long ways to go to know the full story here. Yeah.
Awesome. Thanks, Tim.
Thanks.
I'd like to invite you all to stay and continue the conversation over reception. Tim will be around for a while to answer more questions. Let's thank him again for a really fascinating talk.